Hello, everyone. This is Dr. Houssein Abdul Sater. I'm a physician scientist at the National Cancer Institute. I'll be presenting to you tissue-based biomarkers in oncology. And first of all, let me thank LabRoots for the invitation, and we'll take it from there. I have no disclosures. What I'll be doing today is walk you through a background on biomarkers in general, speak about a few of the tissue-based approved biomarkers by the FDA, and then move on to discuss the tumor immune microenvironment, its complexity, and some of the novel tissue-based biomarker platforms and their challenges, and finally some future directions. First, defining the biomarker, it is a characteristic that is measurable. It has to be measured as an indicator of a process, whether normal or pathogenic process by itself, or as a response to an exposure or intervention such as a clinical trial. And any molecular, histologic, radiographic, or physiologic characteristics are types of biomarkers. What you should know is that biomarker is not an assessment of how an individual feels, functions, or survives. Now, the biomarkers endpoints and other tools resource, which is a resource of collaboration between the NIH, the National Institutes of Health, and the FDA, categorizes biomarkers into several categories as listed here. The susceptibility and risk biomarker are actually biomarkers that would indicate whether the patient is susceptible or at risk of developing a process which is biologic or pathogenic. But diagnostic and monitoring and other biomarkers are really when the patient has an established disease like cancer. A quick brief description of what prognostic versus predictive might mean. A prognostic is an indicator of bad or good outcome regardless of any intervention used for treatment. And examples of that include like the staging, like AGCC staging, the T stage, N stage, and so on and forth. While a predictive biomarker is a biomarker that is used to indicate whether treatment or a specific intervention is good or bad for patients. Now, just an overview of the process that a biomarker discovery and validation goes through. There are different phases that are described in literature, and that varies from one resource to the other. But suffice to say that phase one is mainly the preclinical or the discovery phase or exploratory studies, where phase two is when you have to develop an assay and validate that assay with what we call analytical validity and making sure that this is sensitive and specific. And then moving on to phase three, which is basically clinical validity to make sure that what you're trying to measure is really measured on specific tissues like human tissue, for example. And then moving on to clinical utility, where those different classifications that we talked about come into play, which is basically whether it's going to be susceptibility or risk biomarker or predictive or prognostic biomarker, and making sure they function as they're supposed to be. And the last phase applies to certain biomarkers. Examples of that are the susceptibility or the risk biomarkers, but it doesn't apply to all biomarkers in particular. Excuse me for that. And this is just guidance from the FDA. I do not mean in any way to get you through all the slide, but this is for your reference. Some of the guidelines put by the FDA for, again, IHC-based or immunohistochemistry-based applications. And all I need you to take out of this slide is, that there is a determination of the risk or level of concern by the assay that you're developing, whether it's a class 1 or a class 2 or even class 3. And there needs to be documentation of the specificity and other information that is required as part of the assay that is of interest. I would start with one of the mostly known, over history of the biomarkers, which is basically the human epidermal growth factor receptor 2. It all started back in the late 1980s, and that was started as a matter of fact after a history of looking at tumor necrosis factor, susceptible cell lines, but story told short, that it eventually ended up by looking at HER2 in various ways, main assays including protein, RNA, DNA, and so on and forth. And it was a challenge over the years to apply those different assays to get clinical utility or clinical validity. And after 25 years, in 2007, the American Society of Clinical Oncology and the College of American Pathologists set together a task force to look into these various assays. And what they essentially found that about 20% of those assays were having errors in them or were not accurately reflecting what needs to be reflected. And it only became a good reliable biomarker when that task force took into action, putting together an algorithm for describing what positivity for that assay means. And the other thing that they found is basically the whether you're testing by immunohistochemistry, IHC, or by fluorescent in situ hybridization, whenever you're using a valid biomarker testing under a CLIA certified lab, for example, that the outcome is the same. And the algorithm on the right side of the slide is for breast cancer, is essentially saying that if immunohistochemistry positivity in invasive tumor cells is happening in 30% or more, that is considered positive. Anything less than that is either equivocal or negative. And if it is equivocal, then you could test with the other alternative, which is the fluorescent in situ hybridization. On the other hand, the algorithm says if you're testing with a FISH or a fluorescent in situ hybridization and you had the copy number of the gene, basically of the HER2 gene over the chromosome 17, more than 2.2, that's considered positive. If less than 1.8, it's negative. Anything in between is equivocal, and then you have to use another validated method. In this case would be the IHC. So the message out of this, and I'm sorry if it took too long for you to get you through this, is that not only the validity of the assay from analytical validity standpoint, but also from analyzing the data of how to consider a threshold is positive or negative, and how could that translate into clinical utility using targeted therapies like trastuzumab or other of the newer drugs. Continuing on that, this just gives a summary. This is a paper from OncoTarget in 2017, and the message out of this slide is that really that over the years there have been few IHC markers, including the HER2 that I extensively spoke about, but also things for immunotherapy like the PD-L1 by immunohistochemistry. But the rest of those were mainly done by genetics, whether RT-PCR or next generation sequencing and so on and forth. That just takes you to believe that most of those are one marker. I should say before I move on is that the reason why we started thinking of biomarkers in the world of oncology is that we wanted to move from one drug that fits all to one drug that fits one person, or personalized medicine. And I'll show you later how this actually changed for some of the biomarkers. To be complete, this is just another additional list of the biomarkers, and I don't intend to go through all of them, but this is where I am trying to say to you is that change in the biomarkers, where one biomarker might fit all, which was the basics or the basis for doing studies or clinical trials like the basket trials, is that one biomarker, which is genetic abnormality that is shared among different pathologies or different cancer types, was thought to be the driver behind that pathogenesis of the tumor, and hence it could be used as a target for multiple tumor types. And one certain example of interest is the DNA mismatch repair and microsatellite instability highlighted or bolded here, and also the NTRK fusion that was a good biomarker for specific drugs. In addition, when I was talking about the biomarkers, there are different assays that could be related to each of those biomarkers. But in this case, as an example of a comprehensive genomic profile test, which has been approved over the years for different indications, while it starts with one indication, it might be actually the same testing that could be approved for multiple indications, and this is just a good example of one of those, which is Foundation One CDX, which is approved for many biomarkers, starting with BRCA here, but ending up also with homologous recombinant repair genes, which was just recently approved, maybe last week, for prostate cancer. In addition, shifting gears back to immunohistochemistry and FDA-approved biomarkers in solid tumors using immunohistochemistry, I already overviewed Her2 for you. And then moving on to the other biomarkers, I wouldn't dwell too much onto c-KIT, but suffice to say that it's used for indication with Gleevec or imatinib to treat gastrointestinal stromal tumors. What is more interesting to the field of immunotherapy nowadays is the PD-L1 testing. And as you can tell from this, that there have been at least 3 clones of PD-L1 assays, or 3 PD-L1 assays, which use different clones, the 22C3 from DACO, the SP142, and the 28-8. And as a matter of fact, today it had the FDA approval again for another indication. The message from all of this is that each one of those not only differ by the clone, but also by the way you analyze the assay, and also the what kind of cells you're looking at to evaluate for PD-L1, and also the thresholds. And I'm just going to highlight, one of the first ones that were approved, the PD-L1 from DACO, the 22C3 clone. And this was approved based on 2 major clinical trials that showed survival benefit, whether it's overall survival benefit or progression-free survival benefit, highlighted here by the Keynote 010 and Keynote 025. But all started with the biomarker assessment, phase 1, the Keynote 001. And after showing some benefit for the patients that had at least some positivity of more than 1%, then moved to include basically the enrichment strategy, in which case that patients that had more than 50% or equal to 50% PD-L1 expression on tumor cells, they were included onto the Keynote 25. Moving on to talk about the tumor immune microenvironment. And this is a seminal paper reviewing the tumor immune microenvironment complexity and also beauty, but also the challenges that it poses for us. The way I cut down that tumor immune microenvironment studying from a pathology perspective, but also from an immunological perspective, is that there are multiple immune cells present there. And those immune cells could be looked at as N, for standing for the number of immune cells, how many immune cells are there within the immune microenvironment, but also the quality of those immune cells. And that could encompass many different meanings, which includes function, whether they're immunosuppressive or activating or what so on and forth, but also the intensity of a specific biomarker, in terms of, for example, the expression of coenzyme B or PD-L1 and other things. But the third component of all of that is the architecture. And what I mean by that is where do they belong to, which compartment, which structure. So as you can tell, endothelial cells, for example, are making up the blood vessels, but they're not making up the blood vessels only by themselves, as well as the tumor, whether it's within the stroma or the tumor per se, whether it's at the margin of the tumor or within the center of the tumor. All of those architectural structures are very important to understanding the tumor immune microenvironment. And in addition to that, there are multiple markers that could be able to define any type or any particular type of cell. Now, summary of the challenges with biomarker development in general is for the tumor immune microenvironment is that specificity is very important in the meaning that you need to be specific in which markers you want to use to define a certain immune cell. While one immune cell could be defined by one biomarker, most of those biomarkers are infidel, and in other sense that you need more than marker to define it. And defining phenotype does not necessarily mean function. So myelodeprived suppressor cells, for example, by phenotype, does not mean that they're actually immune suppressive by function. And the reason why we're looking at markers that are static. So the next challenge is that, while we're trying to get some biology understanding, we're only looking at a static view, and those biomarkers are mostly changing over time or even by small changes by preparing the tissue. So perfect example of that is PD-L1, which we explored in previous slides. Other things that include assay limitations is the quality of tissue, and we'll go over those in the challenges in further slides down. The consistency, the reproducibility, and the different antibody affinities. And the perfect example here is the difference in performance, for example, in the SP142 clone versus the 22C3 clone of PD-L1. Now, complexity, what I mean by that is that when you're studying the tumor immune microenvironment, you're really looking at different subspecialties that are put all together. Whether you're looking at metabolism, you're looking at survival, death, or compensatory pathways, all those. And what I mean is by the immunogenic cell death and other pathways, so different disciplines that get into understanding the tumor microenvironment and not just looking at the number, quality, and the architecture of those different immune cells. Now, that being said, I'm just going to start with a success story about the Immunoscore and walk you through that. So the Immunoscore was started as a concept in 2006, maybe before that, by Jean Gallon and his group in France. And they understood very early on the concept that I'm talking about, which is basically the type and the orientation, which fall into the quality of those immune cells, but also the number, which is expressed here by density, and the architecture, which is basically the location. And that consists basically of what the Immunoscore concept was all about. And to simplify that, it basically is a measure of two markers, which is the CD3 for T cells, and the cytotoxic T lymphocytes represented by the CD8 in two compartments, the center of the tumor represented in red as a whole, and the invasive margin around the tumor represented in the dark green color just below the red color. And what it is about is basically taking the densities of the immune cells within those compartments and transforming them into percentiles and taking an average of that. And because you have two markers and two compartments, you end up with basically 4 readings, and you get the average of those 4 readings. And because of the indication initially that this biomarker might stand as a prognostic biomarker, as an independent biomarker, along with the other prognostic biomarkers that were known for colorectal cancer, including the TNM stage and other biomarkers of interest, there was an interest to validate that in an international consortium led by the Society of Immunotherapy of Cancer, and all started in 2012 with about 13 centers around the world, 12 countries, with 3,539 patients, and then dividing those into a training set and 2 validation sets. And story made short is that what the training set showed is basically that, and again, looking at this as an independent prognostic biomarker compared to other already proven to be prognostic biomarkers for colorectal cancers with stage 1 to 3, is that in the training set, the recurrence-free survival was about 8% in the patients. The recurrence rate, sorry, was about 8% in the patients with the highest immunoscore, compared to 19% in the intermediate and 32% in the low score at 5-year mark. And I should say the cutoffs for those immunoscores by percentile was 0 to 25%, 25 to 75%, and 75 to 100%. And this stayed valid in the two controls, the internal validation set and the external validation set. And the main message out of this is that the immunoscore was an independent prognostic marker for stages 1 to 3 colorectal cancer, independent of other prognostic factors, including the microsatellite instability, the T and N and TNN stages, and age, gender, and other factors. But also when they looked into stage 2 alone, that stood valid as well. Now, the Society of Immunotherapy of Cancer in 2006 put together a seminal paper looking into what are the platforms used to evaluate the tumor immune microenvironment, whether peripherally or at the tumor level. And there has been a concept that still is thought of as valid, that are two types of tumors: the immunologically unresponsive, cold kind of tumor, versus the immunologically responsive or hot tumors. And these were defined by various findings from different platforms used. But what stands out that in 2006, that the multicolor immunohistochemistry, and which I will be referring to as multiplex IF, or multiplex immunofluorescence, which takes into consideration multiple platforms which share the same concept, that you have, you are using multiple antibodies or multiple cycles of these antibodies on the same slide, which makes the ability to look into biomarkers that are co-localized or close to each other better or simpler or more effective. And from that, basically, I'll move on to explain what is multiplex immunofluorescence means and why is it important. So focusing on novel technologies, first with immunofluorescence, multiplex immunofluorescence or multiplex IF, I'm going to go over the concept and show you a demo. And in this example here, what we're showing is different cells that have different epitopes represented by the colors, blue for CD20, CD8 with lighter blue or cyan blue, and so on and forth, that will be stained by primary antibodies and then followed by secondary antibodies, which have horseradish peroxidase, that will catalytically make those different fluorophores represented by the various colors covalently bind to the residues around the epitopes that are targeted by those primary antibodies. But the key to this staining method is, which is developed by Perkin-Elmer now, both by Akoya Biosciences, is that the way to get rid of the species-to-species interaction is by denaturing the immunoglobulins or the antibodies, the primary and the secondary antibodies, using heat. And in this demonstration, it's been shown as microwave treatment, but it could be done with various methods that heat up the tissue. And I'll show you the demonstration where primary, secondary, followed by fluorescence staining as a signal detection, followed by the second antibody, the third antibody, and the fourth antibody and the fifth antibody in this case, or the fourth antibody that is shared between these two cells. And this would allow that one cell could be presenting with two different biomarkers that are shown with these different colors, and that would allow to do co-localization analyses. And that goes to the point that there are certain cells that need multiple markers to be defined as a phenotype or a specific phenotype. And this is the outcome of this multicolor IHC. This is one of our studies that we presented at AACR in 2018, and the key point from that was basically that we could push the technology at that time to 8 markers with DAPI, but also we were looking at non-small cell lung cancer changes with demographics, and I'll be showing some of the data at the end of this talk. Now, I mentioned the staining method, but there are multiple ways to stain. The concept is basically that you have tissue represented by blue, you have epitopes, and then you come with the primary antibody or a secondary antibody, and then you have a signal that needs to be detected as a surrogate for that antibody that recognized the epitope. And this signal could be a fluorescence, fluorescent dye, which is light, which will be detected by basically after applying laser light and filters, and that would be the example of the right upper corner detection method. But the other method systems that are shown on the left side, they basically use ion-based signals, and to detect those ion-based signals, you need mass spec to look at the masses of these different oligo elements or oligometals that are used as signals. There are other methods that use other ways of detecting the signal. I will not be going over these in this talk. This is a simple or a simplified or maybe a complex way of looking into after you stained and taking that into a signal detection platform. And as I showed in this example, it's Perkin-Elmer-Acoya's Vectra Polaris scanner, which is shown with the person holding the gloves in blue and holding, the rack with the slides that will be scanned, and you get an image which is shown above that. And what the screen showing is basically you could do segmentation of the tissue after you get your regions of interest scanned again, and you segment basically to walk in the tissue, and then based on DAPI, which is a nuclear marker, we segment the nuclei. And after that, you could stratify or phenotype your cells based on the various markers with those examples shown here in a binary way, in the sense that, whether it's positive for any specific marker or not. But also you get what we call the, again, going back to the architecture, the coordinates in the field by X and Y, and that could be used for various analyses. And the end point of that is basically getting a readout with generation and statistics that you could use to do further analyses. And this is the assay that it was proposed by Perkin-Elmer at the time. I will not get into details, but this is for your reference if you need to go into the details of that assay development. Applications for multiplex IF, there are multiple applications, but the main application that I am very interested in personally, and my lab is interested in, is developing that as an assay for clinical trials, certainly in oncology, but also could be used for other applications, including immunopathology. If you were to think like diagnoses of lymphomas, which would take several biomarkers or several immune markers, and that could be achieved probably with one slide if the assay is developed and validated for that diagnostic purpose. Now, where does multiplex or multicolor IF stand among the various biomarkers used for prognosticating or predicting outcomes to the newer or to the new checkpoint inhibitors or new immunotherapies in cancer? This was a great review, systemic review of the various ways to detect PD-L1, including the multicolor IHC or multiplex IF compared to just the single color IHC PD-L1 testing, the tumor mutational burden represented as TMB, or the genome expression profiling or gene expression profiling with RNA and other methods. And the key message out of this paper was that multiplex IF stands highly sensitive and specific with the highest under the curve area REC curve. And that was a very encouraging paper for the field, suggesting that using multiplex IF is really the way to go compared to other ways of biomarker discovery in tissue level, at the tissue level. And these are just technical overview why multiplex IF is better than regular IHC. The key thing here is that fluorescence uses, with the spectra and the physics of the light, use the ability to detect multiple markers, and the dynamic range changes by up to 2 to 3 logs, detecting basically the intensity. And there is a linear correlation between the intensity of the immunofluorescence and the protein quantity. And there is the chance that you could use spectral unmixing and machine learning with trainable algorithms that could allow for better assessment rather than just a subjective assessment with immunohistochemistry. So that goes back, takes me back to where are we with biomarker development of multiplex IF. I think we're still in phase 1/phase 2 because the platform or the assay is developed, but there are specific biomarkers or biomarker panels that are still exploratory. And the analytical validity is still required, and the community is working on that, and the clinical utility is to be proven. Why is the analytical validity is complex? Because there are multiple steps that need to be validated. I mentioned some of that. One is the staining method used, the signal detection tool, the image analysis platform, the analysis approach, and the advanced analysis after getting the data out of the platform. I'll go through some of the challenges in assessing time or the tumor immune microenvironment. I mentioned those. I'll go over the pre-analytics and staining and detection. And in this example, on the right side or schematic, that gives you a summary of what it takes to go through the tissue processing from the time it comes from the body or tissue in vivo before even fixing the tissue or fixation till the time you do the immunohistochemistry. There are multiple things that are involved, and the big question first, if what we're trying to look at reflect the biological process. Remember, we said biological or pathogenic process that is happening at the tumor microenvironment level. That could be changed by different factors even before getting to the fixation, and that includes, for example, the ischemia from the time the biopsy is taken out or the tissue is taken out to the time that you're fixing the tissue. On top of that, the tissue manipulation, which is basically after the biopsy is taken and you're fixing the tissue, the type of fixation you're using, which is mostly what we use is formalin fixation, and we embed in paraffin the tissue, is that it actually cross-links the antigens or the epitopes. And what happens is that you need to do antigen retrieval methods, and that could be successful or not successful. And if the epitopes are still masked or destroyed by various steps before looking into that with immunohistochemistry, really there is no immunohistochemical staining that will come out of that. And if the epitopes are exposed, there are two things to consider. One is the antibody that is being used, whether it's specific or not, whether it's monoclonal or polyclonal or not. So the antibody itself could be a limitation, if it's a good antibody or a bad antibody, just to put it in simple words, but also how much of the epitope is exposed, how much of the epitope is actually shown up that the antibody will pick, and that will determine the sensitivity and the specificity of the assay. And that needs to be validated, again, to make sure that you got that analytical validity and then clinical validity. And if that is all checked, then you get a good immunohistochemical staining, and that takes you to the other step, which is basically how to read that immunohistochemical staining positivity or negativity. Going through the list on the left I just mentioned to you the pre-analytics. I just add to that with the multiplex IF is you're looking for the antibody fidel. And what that means, not only specific and sensitive, but also because these different immune cells might need different antibodies to define them, most of the antibodies are infidel in the sense they could be shared between different cells. So to define cell A and cell B, if they have shared antibodies or shared epitopes, then you might need different fidel antibodies to define that. And this is where the panel design comes in multiplex IF to make sure that the antibodies that you're using are reflecting what kind of cell you're looking at. And because you're using fluorescence, and this is light, and there's physics to light, and there's some limitations to that physics, is that the signal between the various channels or various wavelength might have an overlap. But I would add to that also that there is some steric hindrance probably between the different fluorophores that are being used, in the sense that one fluorophore, if abundant, might actually block the other fluorophore coming to the same space, where that those two epitopes are sharing the same space. And this is what we commonly call in our multiplexing community as the umbrella effect. And in addition to that, there is another factor, which is time, the time to actually do all this, to do validation, but also the time to actually perform the assay. And some of those assays might take several days if done manually. And you could think of what problems could happen if you're using that time consuming method in terms of reproducibility from one person to the other. Moving on, and just briefly, this is a list of some of the examples of the various multiplex techniques available. And I don't mean to dwell into this, so this is just for your reference with what technology, what signal method detection is used, and so on and forth, and the pros and cons for each one of those. But to brief up on some of that is that those scanners or signal detection, like the Hyperion, which detects basically metal because it is mass spec based, compared to fluorescence scanners, another fluorescence scanner, is that none of those are FDA approved yet, and still they need to go through that. And the only two FDA approved whole slide imaging technologies are from Philips and Leica Biosystems, but they're only approved to do H&D whole slide imaging. But none of the immunofluorescence detection methods or assays have been validated as FDA-approved assays yet. That takes me into adequate representation. So generally speaking, what we do is we take representative regions of interest or fields of view, and typically at 20x or at 40x, and we move on from there to take basically averages, basically representing different tumor areas or different tumor immune microenvironment areas, and so on and forth. And then the question becomes, which is an open-ended question, how many of these FOVs or regions of interest do you need, and whether you're really representing by taking those various randomly selected areas within the specific compartment or specific tumor. And this question was asked by Dr. Rim and his group at Yale in 2011 in breast cancer, and they looked into several breast cancer markers. And in this study, they looked to see whether iteratively, if you were to take several regions of interest at 20x, what would be the optimal number for each of these biomarkers? And you can tell from this figure that varied from one biomarker versus the other. So while cytokeratin, for example, only needs about 3 regions or fields of interest, something like MAP-tau needed like 14, and the average is about 5 to 10 regions of interest. So most of what we do so far nowadays, around 5 to 10 regions of interest, is somehow okay. But most people agree that potentially taking the whole field of view rather than the whole biopsy area or whole slide image is probably better for many reasons. And there was another paper questioning, because we know that there is heterogeneity within the tumor microenvironment. There is heterogeneity within any tumor biopsy. And the question is how, if you were to capture one core, would that be representative? And actually that paper gave a good answer about that, although it's limited by the number of samples that were tested, but basically shows what this paper showed, again, from David Rim's group, is that one core might be sufficient to actually average those differences in the heterogeneity and be representative of, despite the fact that you might be looking, if you were to look at various cores from the same tumor, they will be pretty much similar. So one core is representative, but maybe one region of interest within that core might not be representative given the heterogeneity. So you need to get an average of that, or take the whole slide to reflect on the immune infiltrate densities that are present within that core. Now, excuse me if this slide is too busy, but this is, again, how to give a scoring. So after you after you're going after detecting those immune infiltrates within the tumor microenvironment, there's always a question: how do you sample? What you look for? What do you score? What do you measure? And these are all open-end questions. But this is from the International Immuno-Oncology Biomarker Working Group, putting some guidelines in 2017, suggesting that, and this applies mostly for those images for breast cancer, but there are some intricacies to other cancer types. But I'll walk you through the schematic in the middle. And I'm sorry, I don't have an indication with a mouse, but what you will concentrate is step one is they're suggesting that you select the tumor area, avoiding the areas outside or the satellite areas with the tumor. So just focus on the area of the center of the tumor, and then define what is stroma and what is intratumoral areas, and then scan a low magnification, determine the type of inflammatory infiltrate. What the TIL group is actually, or the working group here, suggests that you only look at the mononuclear stromal infiltrates rather than the areas where you have granulocytes or neutrophils or necrotic areas, which is a valid, commonly accepted concept in among pathologists. And while this is actually meant for a guidance for pathologists to report on, it actually presents some challenges that you have to, if you look at the step 3 and looking how many tumor nests you have and how you want to score the whole area, it's a bit challenging. And pathologists signing out cases every day does not have the ability to go every single case and do that scoring without needing further time and without having actual compensation for the time used for that kind of analyses. So that's what presents the need for artificial intelligence or advanced methods to look at that. And this is a work that is in progress, and there are new papers coming out of the working group on that. Now, the step 5 is to assess the percentage of the immune cells. And as I mentioned, the working group suggests that you get actually various percentages by the compartment, whether it's stroma versus the tumor itself. And this is for practical reasons, because the area that you're measuring might actually dilute the percentage if you were to look at the whole tumor versus stroma alone and tumor alone. And in addition to that, it also reflects what has been looked at by the Immunoscore and by others, is that for certain tumors, you might want to apply also the, and this is the image on the right side, is that you want to look at the center of the tumor, and they define what an invasive margin is here by 1 millimeter. So you draw the margin, and you draw the margin around the tumor edge, and you take basically inside the tumor and outside the tumor a margin of about 1 millimeter. And again, ignoring the satellite areas around that as they don't qualify for the assessment based on this working group recommendations. And I left that there for reference in case you need to go through the details. But one more thing from there is that the threshold for what is called positive or negative has not been defined, and that has been left to be defined by further studies. And it's been suggested that most people would go for an estimate, and that is treated also, instead of being categorical, as a continuous variable, and that might have its own reflections on how to get the assessment done. Finally, whichever method you use, you end up by, you want to analyze the data and make sense out of it and present it. And as a pathologist, for example, to present these different data to a clinician, it might be a challenge. How to understand that, how to represent that, how to make sense out of that, what does it mean compared to traditional ways of doing pathology, which is morphology and giving colors or shades for different things that are looked at from a morphological standpoint, which is basically the grading, or the staging, or the 0, 1 plus, 2 plus, 3 plus method, which is an estimate of things, which is mostly subjective compared to an objective method of getting data. And these are the different image analysis platforms. I've mentioned before that we do unmixing using algorithms to unmix the different spectra of the light. But after you get that unmixing of the various channels, you get to do the analysis on the images, and that might have different platforms available for you to use. And then becomes, if you're doing this as an assay, which platform do you use? And I just put in there some of the platforms available for your reference. So for the analytic approach, I think, and this is a key thing, is what is the key question when you're trying to analyze the tumor immune microenvironment? I have my own concept on this, that the tumor immune microenvironment is really, and that was basically the reason why I'm putting this message out there for you. In my mind, I was thinking of an analogy like the Facebook, face recognition algorithms, which would detect a person A with a picture 10 years ago compared to a picture from now, and would say, this is person A, this is person A. And then it would detect whether the patient is smiling or frowning, and so on and forth. And then I said, the tumor immune microenvironment has patterns that we recognize as a morphologist, as a pathologist, but how does that reflect? Can we actually detect that? Can we? The first question I got from my collaborators and colleagues from artificial intelligence world is, do you really believe that the information is there? And then the very next question, where is the message? If there was a message, because my answer to that, yes, probably yes. But that put me into different questions down the road. These questions bring into questions, and this is very important when you're planning for any kind of analyses or trying to discover a biomarker in a specific field like oncology. And when you're doing all of these analyses, you want to see whether the pathway that you're taking to get the data or to do the analytic approach would actually give you the answer. And one major question is, if you were doing batch analyses and small batches, you'll get probably some accuracy, more than 90%. But when you go around that training algorithms and you want to do batch analyses, most of the time, my observance and many other groups' observance, that the batch analysis would fail you to do any accurate analysis more than 80% or even 70% of the time. And the question is, do you really need to be subjective in terms of doing that, or you want to be just let the algorithms do their job? And then that takes you to the next step. Do you want to use thresholds? Do you want to be subjective, or actually train those thresholds to define what phenotypes are? And then the other question would be, are you counting, or you want to count? You want to have a number of cells, or you just want to get a density, like basically looking at a concentration of a solute in a fluid, and you just don't want to know the number of the solutes. You just know whether it's concentrated or not. And also, do you want to do compartmental compartmental analyses or not. You want to look into tumor versus stroma, core of the tumor, and base margin. All those add to the complexity of your analyses and how do you want to present that. In addition, I spoke about patterns and, talking about pattern recognition as like face recognition for me. And doing spatial analyses is just part of that. And the question again is whether you want to let the algorithms or the neural networks that you want to train potentially do unsupervised analyses and dissecting the tumor microenvironment in a way that you could tell whether there is a pattern there or not. And after all, there is no given statistics for all what you are doing. It's just going to be on the way, depends on the way that you're doing your analyses. And finally, after all, I presented you with a lot of challenges, but I think after all, I think there have been multiple data out there suggesting that there are multiple factors that might affect the time or the tumor microenvironment, and not only the tumor itself or the immune cells themselves, but those could be affected as other things in the human being or the biology of the human being could be affected by multiple factors, and those are known. If you're coming from Africa or you're coming from a different country, that there is a variability in the MHCs, there's a variability in multiple immune factors based on age, gender, race, medications, comorbidities, genetics, epigenetics, and the environment, whether food, location, stress, etc. And one of the things we were interested in, because in lung cancer, we found, looking at the SEER data, for example, that the natural history of the disease varies by gender and race. And does that actually reflect into the tumor immune microenvironment? So in a study that I showed you the images earlier from AACR 2018, we just looked into a small set of patients that were selected to be similar. They're all smokers. They're all sharing the same characteristics from a pathology perspective. But the basic question we wanted to know is, do these immune microenvironments change or vary by gender and race? And not to our surprise, because we were expecting that, is that these immune cells were actually different between males and females, and that actually went along with the natural history of the disease. Basically, that males did better than females, and that blacks did, and they make a difference for how they by themselves for the race, but maybe if you put the two together, it makes a huge difference. And what was important, which I'm not showing here, is how those immune cells were actually excluded in the black race, although they had more immune cells compared to Caucasians or white by reported race or reported color. Although they had more inflammation, but maybe they were, immune excluded or immune suppressive. And that just highlights the fact that the immune microenvironment is not just complex by architecture, number, and quality of these immune cells, but by the other factors that I just mentioned. Now, can we do better? And this is the ABC, or my APC in my lab, which is basically the automated panel concept. I think that if we were to automate everything in those assays, we could do better. And that would be starting with the panel design, figuring the puzzle of how to put these immune globulins or the antibodies in tandem, to using the fluidic robotics to get the dilutions of the various reagents, to fully automated staining using different technologies, and then moving on to scanning the whole slide and figuring out an algorithm how to do the analyses. Even that validation would take us to basically the artificial intelligence and methods how to do that. After getting the data or the images and then doing the analyses. Now, automating any of these steps is a major work to do by itself, and it requires basic quality control and validation. So the complexity here in doing the multiplex IF is that you have the various steps, from the staining to the dilutions to basically knowing what antibodies you want to use and how you want to use them, and then the scanning process. And if we were to go for an FDA approval, I think that any of those need to be comprehensively validated, and that includes, as I said, the ability to do the analyses on these data. And I'm sorry for this slide. This was green in background here, but it does seem to be changed. Now, that takes me to the take-home messages on multiplex IF. What I think of multiplex IF, this is a powerful tool to dissect the complexity of the tumor immune microenvironment. It's a new school of biomarkers, that biomarker discovery, actually, that bridges multiple disciplines. It's just not pathology. It is actually pathology, biology, chemistry, physics, bioinformatics, data science, immunology, oncology, so on and forth. As long as it's the beauty of that platform, it is actually the challenge that you want to be knowing that all of those play a role in determining your final outcome. I think global automation is one way to improve reproducibility, quality control and validation with the use of stringent laboratory accreditation standards, proficiency testing, and competency assessment is essential. That takes me back to the Her2 story. And consensus algorithms that rely on accurate, reproducible assay performances are needed. Monitoring and repeating testing in CAP-accredited, CLIA-accredited laboratories are definitely necessary. With that, I would thank you for watching my talk. And if you have any further questions, please feel free to contact me. I left here on the left side all the social media contacts. And I should highlight that we started last year a scientific interest group for multiplexing. It's still in its infancy, and the website is under development, but I left it there just in case you're interested. You could get there to that website and subscribe, and I would be happy to provide you with any resources or information that is needed. And thank you.